Hello all, this is Kristen Haberther welcoming you to this Bite-Sized Bio webinar, which today is sponsored by Astarte Biologics. Astarte Biologics offers the largest selection of characterized immune cells and reagents to help immunology and inflammation researchers get on with discovery. Astarte makes it easy for life sciences and pharmaceutical companies worldwide to find the right cells for their research needs. Today's presentation is titled The Mysterious World of Macrophages, How to Harness Them for Your Research, and is being presented by Anne Lodge from Astarte Biologics. Anne Lodge is the CEO of Astarte Biologics, a supplier of high-quality immune cells, reagents, and research services. She holds a PhD in cell and molecular biology from the University of Vermont and has an extensive background in cell-based therapeutics and immunology, including multiple patents. As always, we will have a question and answer session after the presentation, so please type in any questions that you have into the questions box, which appears at the right-hand side of your screen, and I will put them to Anne at the end. So the recording of the webinar will be available on our website in about 24 hours. So now over to you, Anne, for the presentation.
Thank you for that introduction, and welcome to the mysterious world of macrophages. I'll attempt to give you a little bit of an introduction to what these cells look like and how to culture them and what you can make them do in culture. So macrophages, they've been described for a long time. They were first described by Mechnikov in 1882. He had a little organism that got a thorn stuck in it, and he could see these great big cells that were eating things. So the very aptly named macro for big and phages for eating. And they're large cells compared to other cells in the circulation, and they ingest stuff, just almost any little bits of things that are around, whether it's bacteria, yeast, pieces of other cells, whatever debris. They are the janitors. There are numerous types of macrophages, and just to confuse matters, they've been given some different names. The tissue-resident macrophages in some organs are really described separately. For example, in the brain, there are microglial cells, which are really kind of the macrophage of the brain. In the liver, they're called Kupffer cells, and in the skin, Langerhans cells. And I think there's some in other organs as well. These are an interesting kind of group of macrophages because recently it was shown that these all develop from cells derived from the yolk sac. Very elegant study showed that early in embryonic development, these macrophages exit the yolk sac and populate various organs, and they stay there for keeps, pretty much. So they have a different life history than the passenger or transitory macrophages, or what we just call macrophages, which are derived from the bone marrow along with the other blood cell types. And then they kind of migrate around the body. They might pass through the brain, they might pass through the liver, but they don't necessarily stay there. The other types of macrophages that are of interest these days are categorized more on their function. So M1 macrophages are kind of the more traditional macrophage that we think of responding to an infection and making cytokines to kind of, so they're involved in that inflammation response and clearing up some insult to the body. The M2 macrophages are more involved in the healing phase of that, or any other insult or injury, and produce less inflammatory proteins and more proteins that are involved in actual tissue regeneration or healing. These are also associated with tumors. You will see references in the literature to myeloid-derived suppressor cells, MDSC, And these can be thought of as dendritic cells or macrophages that have kind of reverted to this phenotype that limits inflammation and tamps down the immune response. And that's part of the reason that there's interest in signals that might drive macrophages to join one or another camp of the M1 or M2. And honestly, it is more of a continuum in terms of, you know, these two are described as extremes, but you'll find macrophages kind of blend into each other at some point. And before we dive in any deeper, what the heck are monocytes? Well, monocytes are in the peripheral blood, and that's the only place you find them, because as soon as they go into the tissue, they're called macrophages. So you don't find any macrophages in the blood, you don't find any monocytes in the tissue. It seems to be just a naming difference as to where we find them. Monocytes in the blood, exit the blood vessel, and then we consider them macrophage, and they kind of replenish those passenger or transitory macrophages I mentioned a couple slides back. So with human cells, commonly research is done on monocyte-derived macrophages, and we'll talk about that a few slides further on. Which is where the meat of this is culture of macrophages, because we do get many questions about this. One of the first things you need to think about is what you're going to culture them in. And you have plenty of choices out there. There are serum-free formulations on the market. There are sort of your traditional classic media that we're all familiar with, like RPMI and DMEM. And then there's also supplements. You will need to use macrophage colony stimulating factor for your macrophage growth. So this is also known as CSF1, colony stimulating factor 1, or macrophage colony stimulating factor. It is possible to grow mouse cells in human MCSF, but I don't believe it works the other way around. So there is some cross-species reactivity of this cytokine. Easiest thing is to just get yourself some human recombinant MCSF and use that. And under those conditions, you can pretty easily grow mouse macrophages from bone marrow cells, the progenitor cells in bone marrow. Put those cells in culture with MCSF, and away they go. And this is a beautiful example of what those cultures look like. There's sort of a variety of morphologies here, but, you know, kind of stretched out, some very elongated, some more flattened out. This is a beautiful little culture going here. Well, we wanted to know what the best medium was for culture of our mouse macrophages. And so we tested some classic media supplemented with fetal bovine serum and varying concentrations of MCSF. So you can see in the legend that we tried RPMI. RPMI supplemented with hypoxanthine and thymidine, which we found is just kind of a nice supplement for rapidly growing cells. Icecove's modified Dubalco's medium, and DME F12. So 3 media formulations that you can buy from just about any supplier of tissue culture medium. And what you can see here is we did test different concentrations of MCSF, and we measured the number of cells present using cell titer glow, which gives a luminescent signal that's proportional to the amount of ATP present. And the amount of ATP is proportional to the number of cells. What is clear here is that the IMDM formulation seems to support growth better than the other 3 formulations. Maybe DME F12 caught up to it here at the highest concentration of MCSF. RPMI, which is a very commonly used medium, especially in immunology labs, clearly isn't as good as these other two. I mean, they're probably all equivalent down here, aside from IMDM, but not so much at the higher concentration. So things are different when you get to the human macrophage culture. So traditionally, monocyte-derived macrophages, as I mentioned, they take monocytes and culture that with MCSF. But monocytes aren't progenitors. They're already differentiated, so there's not a whole lot of growth that goes on in that case, where with the bone marrow progenitors from mice, you can grow a lot of cells. We've also found that monocytes that we prepare by negative selection may contain a small number of CD34 cells. So if there is proliferation in a human monocyte culture, it is probably due to that contaminant, if you will. So similar to our mouse system, we tested, in this case, a serum-free formulation, ex vivo 15, with MCSF, and the two more traditional media that were good for mouse, DMEF12 and IMDM, also supplemented with MCSF and with human serum. So just to show you some of the morphology that that we saw and that you might see in some of your cultures, this is in the serum-free ex vivo 15. And these long, delicate cells are kind of classic macrophage. You can see this blue arrow pointing to a really long and skinny migrating macrophage. And it actually at the other end of the arrow is another thing I usually call a fried egg kind of morphology, because it's a big splat of cytoplasm with the yolk in the middle being the nucleus. And what you see a lot of in this field and in ex vivo 15 are cells that are not attached to the plastic. And that's pointed out here by the yellow arrow. And all of these really bright cells are cells that just haven't attached to the plastic. So it looks very different in the presence of human serum and DMEF12. You can see that this is pretty much confluent, for one thing. There's a ton of cells in here. And I got to say that this is the same number of cells from the same donor, but just cultured in DMEF12. Many more of these round, flattened out cells. Most of these appear to be attached. Here's another one over here. But all through the field, you see these more rounded cells, less of the elongated type, although there are elongated cells in here. They're just less frequent. And then sort of in between is the IMDM formulation, where there's a mixture of the elongated cells. You can see their little skinny little processes sticking out, but also rounder cells of the more fried egg type morphology sitting around in here. So then, we had these nice morphology differences to look at. We also wanted to see what's the function like. So we stimulated those cultures with lipopolysaccharide endotoxin, if you will. And this is a strong activator of macrophages. And it was a really big difference between the three medium. Cells cultured in DMEF12 produced much more IL-6 and TNF-alpha compared to those same cells grown in ex vivo or IMDM. So for instance, with TNF, it was nearly 9,000 picograms per mil versus less than 1,000 for the other two media. What was interesting is if we stimulated them with poly-IC, which binds to a different toll-like receptor, TLR3 versus TLR4, now it was ex vivo 15 that supported better cytokine production. DME F12 still a pretty good second, but IMDM not at all, or much lower levels than the other two. So something about ex vivo 15 perhaps supports more expression of that TLR3. It's hard to know, but an interesting difference in cytokine production among these three media types. One other thing to consider when you're culturing your macrophages is that they adhere to the tissue culture plastic very, very tightly. You can scrape them off, but anytime you scrape off cells, you will have cell damage and death. You can't get them off using trypsin. You might be able to get some movement with acutase and maybe with EDTA, but even those two compounds, very difficult to retrieve those cells off of the plastic once they've decided to stick. And just to show you a comparison here, if you culture your macrophages on ultra low attachment culture plastic, you can see that these are all really bright and round, as I showed in the earlier photomicrographs, with only a few of these long stringy cells attached. And the same cells on tissue culture plastic, you can tell, are very much adherent. So our recommendation is that it's best to culture them in the configuration you will use them. So if you're going to do something in a 96 well plate, put them in there and be done with it, because it's really hard to get them back. The second best thing you can do is use ultra low attachment culture vessels from Corning. They are kind of pricey, so you really need to be convinced that you need to recover them after your culture period. A cheaper alternative is non-tissue culture treated plastic. You can use bacterial petri dishes that are not treated for attachment of cells. And those are better, but it's still a lot of work to get the cells off. So again, really recommend putting them in place and using them as is if you can. So finally, a little bit about M1 and M2 macrophages. So as I mentioned before, M1 macrophages are sort of our classic, they go along with the Th1 inflammatory phenotype. They make a lot of pro-inflammatory cytokines, present antigen, and produce nitric oxide. And the M2 are more making IL-10, IL-13, not many inflammatory cytokines, and not making nitric oxide, and they build up your extracellular matrix to kind of knit things back together. So the conditions that favor one or the other, if you grow, as I've just described, most cultures involve using MCSF, but that favors an M2 phenotype. So if you want to switch them from an M2 to an M1, you can treat with interferon gamma, which will skew them towards M1, or with GM-CSF, which is also more of an M1 type cytokine. Stimulation with LPS very much favors that inflammatory cell type, as it makes biological sense that it would, because it represents a bacterial structure that macrophages are designed to recognize molecular patterns of pathogens, and this fits right in with that. Stimulation with poly-IC may also have that same effect of switching things to an M1 phenotype. And this, these two photomicrographs give you a sense of what happens to your macrophage after activation. The control macrophage here have more of those rounded shapes, looks like this is in DME-F12, but kind of chubby little cells. And 48 hours after adding LPS, you'll see very long, extended, stretched out cells. And anytime you see a culture full of these, you know that there's been some activation going on, and there's probably going to be cytokine in the medium. So macrophages are a really versatile cell type. We haven't spent a lot of time talking about some of the other things you can do with macrophages, but they have some great tricks like phagocytosis. Of course, they can migrate. That's one of their things. But they are difficult to work with in the lab because of that tight sticking to plastic. Selection of your culture medium is really important. It will influence function of the cells and your ability to measure activation. So take the time to learn what works best in your laboratory. If you do use serum supplementation, that will have an impact as well. And make sure you set yourself up for successful experiments down the road by kind of checking your conditions before you dive into the meat of your experiment. And thank you for your time and attention. And we can take some time to take questions and discuss. Thank you very much.
Thank you for that really interesting talk. And yeah, that was really interesting. We have some questions from the audience. So the first is from Martin Johansson. He says, from my associates, I've heard that human macrophages can only be cultured in human serum. Otherwise, they senesce and or die. What are your thoughts about that?
That hasn't been my experience, but that might be because of a lack of MCSF that is species compatible. MCSF would be present in serum, but if bovine MCSF doesn't work on human cells, which it may not, yeah, then you're out of a key growth factor.
Yeah, absolutely. Yeah, I completely agree. I think even for a period of time, we were concerned in my postdoc lab about um, negative effects of serum. And so for a period of time, our macrophages are being grown in serum-free media, and they were still fine. So,
Right.
okay, so next question is, if most residential macrophages are M2 and express CD206, will targeting it pose global toxicity?
Good question. Actually, I would think you would get, it wouldn't be targeted to a particular organ, that's for sure.
Yes, absolutely.
But keep in mind that a lot of the bisphosphonates that are prescribed for osteoporosis, their whole mechanism of action is dependent on killing off macrophages.
Right.
So there's that.
There's that.
You can live without them for a time, I guess is what I'm saying. Yeah.
Yes, absolutely. Okay, so what about another attendee is asking about the dose of LPS that you used for your macrophage cell culture.
Oh, we usually use around 100 nanograms per mil.
Okay.
And we have titrated that. It's always worth doing a titration when you're working with almost anything. And there's a wide range of quality when it comes to LPS. But a good clean prep would actually give you some effects, even down at a nanogram per mil.
Do you, this is in no way a product placement, but do you have a particular resource that you usually go to for your LPS that you could suggest?
We honestly have been getting it from Sigma, but we do another purification in-house. because it's really hard to get LPS that doesn't also have lipoproteins present
Sure. Sure.
or lipopeptides that would then bind to other toll-like receptors.
Right, right. So you just do a purification at that step in house. That's cool.
Right, right.
That makes sense. Okay, moving on. How does the addition of GM-CSF affect the maturation of MDM?
That's a good question. And that's some of what we were playing around with when we were looking at different culture media. And it's a little bit of a tricky question because for some of us, we're used to thinking of growing monocytes in GM-CSF gives you dendritic cells.
Mm-hmm. Mm-hmm.
I think this is sort of just a name difference,
Yeah.
put it that way. If you do MDM in MCSF, like we usually do, and then throw GM in later, you'll get more of an M1 type response out of those cells.
Mm-hmm.
Yeah.
Yeah.
So I think, and for some people, they would grow in GM CSF and still call them macrophages.
Yeah.
So yeah.
Yeah. It's just kind of what your categories are, what you're
Yes.
specifying. Yeah.
Depends on what you want to call them.
Right, for sure. Okay. The next question is Jennifer's asks, I use recombinant human insulin in my monocyte to macrophage differentiation cultures. This works well. Do you think
Uh-huh.
this pushes them to M1 or M2?
Ooh, got no idea. That's that's a you should check. I would
I was going to say
my
this sounds like a thesis project.
that sounds like something to look at. Well, and there are she's probably more aware of this than I am, that there are obesity effects, metabolic effects that maybe skew things more to an inflammatory state. So maybe
Hmm.
insulin would do that. I don't
Right.
know.
And maybe even just
Yeah,
at a certain concentration too.
sure.
Right.
Yeah.
Yeah. Who knows? You're going to have to.
Insulin has funny effects in vitro because it can, I think it can also act through the IGF-1 receptor.
Oh, I didn't know that.
I think.
Oh, okay.
Don't quote me on that. It's just, there's a reason that's called insulin-like growth factor.
Got it. Fair enough. Okay, what is this next question? This is a good one. What is the role of M2 macrophages in tumor progression?
So yes,
I think this,
M2s are kind of
yeah,
figured on being more suppressive-like.
yes, and I think perhaps they're asking about like TAMs.
Yes,
Yes,
tumor-associated macrophages
right.
or myeloid-derived
Mm-hmm.
suppressor cells or
Yeah,
that that whole fuzzy group of cells that are found in tumor microenvironments and may play a role in screwing up the possibility of getting rid of tumor cells.
that's right.
Yeah, exactly.
Mm-hmm.
So if they're not pro-inflammatory, they're a problem, and we
Yeah,
should do something about
yes,
that.
you should wake those up. Um, for supplements, is low or high glucose required to grow them in culture media?
The formulations of DMEM we play around with are usually the high glucose
Okay.
formulations. I don't know that the high glucose level is required
Why is it that you guys use high
because we haven't
glucose?
honestly compared the two formulations.
Oh, okay. Is it just from a previous protocol that you use the high glucose, or just
Yeah, I
okay?
think so. I think I tend to get that kind,
Mm-hmm.
figuring that for rapidly growing cells, the high glucose is beneficial. But
Yeah.
honestly, you could certainly take a step back and say, well, there's DMEM that's got, you know, I think it's a gram per liter of glucose, and then there's the 4.5 grams per liter.
Mm-hmm. Mm-hmm.
Big difference. So
This is going to seem this is my own question as we're talking about this. When with the high glucose and the macrophages, with the high growing macrophages, do you think that that could put them into a different metabolic state and then a different differentiation pathway than otherwise?
Um, it could.
Mm-hmm.
It could, given all of the metabolic um, studies that are coming out.
Mm-hmm.
Um, you'd have to wonder about that.
Yeah. Okay. So Hisham asks, how many macrophage passages can we do?
You can't do any with with human macrophage.
No.
Sorry, the human macrophage just don't divide. That's
No.
it.
No. They're
And the
not
only growth
dividing.
they're going to get is if there's CD34s in there. But with the mouse, they seem to keep on going.
Hmm.
Of course, you're starting from progenitors,
Well,
and
okay.
that's probably some of the deal, is that you take bone marrow stem cells and get them dividing.
Right.
Um, yeah, those go for a while, but they're kind of funny as far as passage goes because as I was explaining to someone last week, you kind of wait for the cells to pile up on each other, and then you take the ones that are kind of on top
Right.
and pipette them off and move them to another flask.
Mm-hmm. Yeah, then you can use those
And you can
ones.
keep doing that for weeks.
Interesting. Okay. So why are macrophages important to study cancer cells? And then, so this is two questions. So that's the first question. And the second is, MCF is the biomarker for which disease? I feel like this is a test.
Oh, that is. Um, so so the macrophages are important to immuno-oncology because
Mhm.
of what we were talking about earlier, that
Right.
if they're not in, they're probably a problem for people working on therapies that rely on the immune system to attack the cancer.
Right.
Um, because they can dampen down that response, we want to understand what moves them from one direction to the other. Um, MCSF. Oh, so
Oh,
there is a
yeah.
mouse that is an osteopetrotic mouse, and I can't remember if it lacks the receptor. I think it lacks the receptor
Mm-hmm.
for MCSF or CSF1. And without that receptor, they are they basically are born with osteoporosis,
Gotcha. Huh. Curious.
which kind of doesn't make sense, does it? Hmm.
Mm-mm. Yeah.
But
Well,
anyway, yes,
yeah.
there are mutant mouse strains out there that have a disruption in that whole circuit.
Okay. Okay.
And what's highlighted is the effects on the skeleton because
Yeah.
they are involved in tissue in remodeling bone.
Macrophages are.
Yes,
I didn't know that.
under their code name osteoclasts.
Oh, duh.
That's just,
Okay. Right. Yeah.
yes, I know. See, we call them something different, but
And then
honestly, they're
you put them in a whole different category. It doesn't even make any sense. Okay. Wow. So curious. Okay.
there's the resident macrophage of the bone. It's
Right.
called osteoclast.
Right. Duh.
Uh-huh.
Uh, I always just, whenever,
Yeah.
as an immunologist, you just think of like Kupffer cells and, glial cells, and you don't, you don't always think about that. Oh, okay. So, do antibiotics affect macrophage growth?
Um, if you use enough of them, sure. Um, depends on the antibiotic and all of that. I'm just not a big fan of using antibiotics in tissue culture media because the
Hmm.
world is just full of too many antibiotics.
Yeah.
Um, so that's that. GM gentamicin will inhibit protein synthesis in mammalian cells if you crank it up high enough.
Uh-huh.
Um, so that's one that people still use,
Right.
and at a certain concentration, it's probably not harming the cells a bit. Penicillin probably doesn't bother them, but it doesn't bother anything on the planet anymore because it's,
Exactly.
everything's resistant, except maybe some streptococci.
Yeah.
Um, yeah.
Yeah. Agreed. Um, okay, what do you think of using MEM for macrophage culture?
Um, MEM probably is pretty limited in its nutrients. That would be my guess. Although I think I've seen alpha MEM used for osteoclast culture. So, so it might work, especially if you supplement with serum. I think serum kind of covers over all of the defects in any particular formulation
Mm-hmm. Um,
to some extent. Just,
yeah. Yeah, to some extent. So this is two questions as well. So, Michael says, in using low attachment plates, the morphology of the macrophages are different. How does this affect any functional readouts? And would you expect the macrophages in a low attachment plate to behave significantly different than in normal culture plates?
yeah. Well, so they might behave differently because, the whole thing of macrophages sticking so tightly to plastic and not wanting to move.
Mhm.
Um, I know colleagues and I once had a discussion and we proposed, though we can't really prove it, that the macrophages are actually trying to engulf the plastic.
Huh.
They recognize it as a surface that's not normal, and so they're trying to phagocytose something that's bigger than themselves. So that's got to do something to them.
Yeah.
Um, and I don't know how the low attachment stuff, honestly, I don't know what the surface modification is.
Hmm.
Um, Maybe that's better for them because they're not trying to respond to something. But I haven't honestly looked.
Fair
It's
enough.
more of a practical consideration of, gee, if I want to work with these cells, I need to be able to move them from place to place.
Gotcha. What is the difference of using spleen versus bone marrow for macrophage prep?
Ah, from mice. So, um, I haven't done this in a lot of years, but I think I was able to grow a macrophage from spleen. The nice thing about bone marrow, though, is that you have progenitor cells there.
Right.
So they're much younger in their development, and you probably get more cell division out.
Mm-hmm.
Um, yeah, when we phenotype spleen cell suspensions, there's not a lot of macrophages there to begin with.
Right.
Um, yeah, that's all.
Yeah. Just different areas.
And you'd probably be taking the macrophages that are there and just keeping them going versus the bone marrow where you've got progenitor cells and you're driving their differentiation to macrophage.
Yeah, exactly. Um, this kind of goes back to the other question, but they're asking, is it necessary to add antibiotics, i.e. pen strep, to your culture media?
No.
I would have completely 100% agree with that.
No, in a word, you do not need those things.
And so I used to, my postdoc was in a laboratory in Seattle where we were doing a lot of kind of preclinical stuff. And so, aside from just that we don't need it, is the fact that if you do have it, it often masks any kind of infection that would be present in the culture media, like mycoplasma, that could alter and affect all of your growths and your readouts. And so it's much better to just know straight ahead if there's some kind of um, contamination than to put in some kind of antibiotic to make
Right.
your culture
About
look
the
pretty.
only thing I can think of is, whether you're collecting tissues from mice or from humans,
Hmm.
there might be some value to including antibiotics just in sort of transport media,
Sure, but in culture
just so
media,
that if anything's in there, you've kind of prevented it and you can probably, you know,
yeah,
lose it along the way. But for routine tissue culture, we really shouldn't be using that stuff.
yeah, Yeah. Um, let's see. How did you ensure that the LPS effects were stable during the experiments? Maybe like reproducible? Or this says how are you sure or how are you sure that the LPS effects was stable during the experiments?
Hmm. Not sure I
Yeah.
understand the question.
Miriam, if you wouldn't
I mean,
mind.
we added in LPS and we just leave it in.
Right.
So,
If you're still here, Miriam, if you would go ahead and maybe add an additional question for clarification, and we'll come back around and hit that again. Okay. Oh, Jennifer was saying, ooh, interesting about the diabetes aspect. She was the one that asked about the insulin question before. Hazel asks, what are the best conditions for getting THP1 cells to produce TNF alpha?
huh. Gee, I haven't had any trouble with them doing that. They seem pretty capable.
Yeah.
Um, yeah.
Maybe check your
Um,
cells, the, s- the THP1s.
yeah.
Yeah.
The THP ones should respond pretty well to LPS stimulation
Maybe, yeah. If they're
and
not,
make TNF alpha.
yeah. I wonder if they've just been passaged too many times, or because you can passage THP1s, right?
Yeah.
So maybe there's just a high passage
Oh
number.
yeah.
Yeah,
I wouldn't think so, man. These are transformed cells that have been in culture for God knows how long.
sure.
They're a leukemia.
Right.
So they just keep going. Yeah.
Oh, maybe check the cell
Yeah.
source.
Although, yeah, I'm not sure. So if they're not responding to LPS, are they in a serum containing media?
That
I
could
haven't
block it.
honestly looked to see if they express CD14, but they are usually cultured with fetal bovine serum and then, yeah,
Yeah. Hmm, that's curious. Okay.
yeah.
Um, do you have experience in co-culturing macrophages with other cells, such as cancer cells, fibroblasts, etc.? And what is the potential for artifacts?
Huh. Um, yeah, we've usually done it deliberately when we do the co-cultures. Um, so we've done co-cultures to see macrophage phagocytose cells. So you'll
Mm-hmm.
see that.
Mm-hmm.
Like you might lose your tumor cells um, to the macrophage.
Right.
Um,
Which you should.
and right, which you should. And we've done antigen presentation experiments and looked at cytokine production and had the question come up, well, who's making the cytokine? Is it the macrophage or is it the T cell?
Right.
And you can do intracellular cytokine staining to kind of satisfy yourselves as to who's making what
Mm-hmm. Mm-hmm.
in those circumstances.
Right.
Yeah.
So Miriam got back about the LPS question, and she says that
Huh,
she
okay.
has the experience that the same dose of LPS from the same drug batch has had different effects on animal health scores. So I think she was wondering then
In,
back to your experiment, were you sure that it was consistent across the board?
oh, yeah. Okay. So is that just a sample experiment or would that happen
Right.
over and over again?
Yeah,
But yeah, I mean, that's meant to be representative of several experiments. Um, and certainly the amplitude, the magnitude of the response varies.
yeah. Mm-hmm.
And that seems to be just a feature of cell-based assays
Yeah.
that we have to live with. Um, and if she's talking about doing experiments in mice, I would think the same might be true. Um,
Yeah. I mean, even
one would hope it wouldn't be too different, but
yeah, but you'd have some kind of a difference in responses. They are different animals still.
right, because of the differences in the mice themselves, not necessarily in the LPS.
Right. Exactly.
whether they're under stress or
Mhm.
are they a different age or
Right.
did somebody change their feed or their bedding. It's very strange with mice, small things can make a difference in their immune response.
Mhm. Just because of the small environment, the small body.
Well, it's things that we don't think are important are apparently important.
Mhm.
I can only say that um, when I've worked in mouse models, specifically I've worked with the EAE model,
Mhm.
and if you move to a different institution and you try and do it the same way,
Mhm.
you can have difficulties.
Yeah.
And we did, and we tried different bedding, and we tried mice from different suppliers, and we checked into the chow and all of these things.
Mhm.
So yeah,
Yeah. Stressful.
funny
Uh,
how that works.
it is,
Yeah,
yes. Why won't everything just stay the same so we can
really
make our
bad when you're trying
experiments,
to complete a postdoc and the mice aren't getting sick.
right? Really need that standard deviation to be a lot smaller. Thanks, guys. Um, so
Yeah.
can both M1 and M2 become osteoclast?
Oh, probably. I mean, there's certain growth factors to drive monocytes to differentiate towards a osteoclast
Yeah, I think it.
phenotype.
Mhm.
Yeah,
Yeah, it's just more of like M1 and M2 are supposed to be easily identify like inflammatory versus more suppressive, but it's the location that calls them osteoclast versus whatever, microglial
right,
or yeah,
right.
that kind of thing. Okay, oh man, this I worked with people that I wasn't actually working on the macrophages in my postdoc lab, but was a big point of discussion in our lab, almost every lab meeting. What markers would you recommend for the distinction of M1 and M2 macrophages, and are there different markers for TAMs?
Um, I think there are some publications out there on it, and I haven't kept up with my reading well enough to tell you what they are. I
Yeah.
just know I've run into some people claiming different expression on M1 and M2.
Hmm.
To me, it's more of a functional thing than a surface marker thing. But then again, there's always a surface marker for something.
Yeah, which I think can kind of get a little complicated too when you're trying to discern
Yeah,
everything because you can have it's not just like no there isn't going to be any 206 here, it's there might just be a little bit less than, it's all kind of on a spectrum rather than a quantitative yes and no, or qualitative I should say.
right.
Right.
Yes,
Yeah.
right. It is a spectrum. So,
Hmm.
right, where are you going to draw the line and say they've crossed over
Yeah.
based on expression of 206 or anything else for that
Right.
matter?
Yeah, exactly. Okay. Let's see what we have. Yes, this is true. There are monocyte subtypes also, classic, intermediate, and non-classical. Oh, which one gives rise to M1 or M2 phenotype of macrophages?
Oh dear. Now, see, when the audience gets smarter than me, I'm in trouble. Um, because before before today, I
Hmm.
was reading something where right off in the intro somebody said, "Well, people used to think that monocytes became macrophage, but no." And it's like, wait a minute, wait a minute.
What?
Um, so I would turn the question around since I don't have a good answer. Um, what distinguishes a monocyte from a macrophage?
Hmm.
Really? I mean, I would like somebody to answer that for me. I've given my answer in this talk as being, monocyte just happens to be in the circulation.
Right. Yeah, that's what
And
we're taught.
once it exits the circulation, we call it a macrophage.
Right.
Um, Nobody refers to macrophages in the circulation.
Right.
But if they're changing that definition, somebody needs to catch me up.
Fair enough. we'll send you back. Um, isn't
Send
it?
me back.
Yeah. Yeah. Is it? Yeah, I can't remember. This is so long ago. I'm not even gonna talk. I'll be quiet about it. I just remember the, because do monocytes, don't have as much of a phagocytic functionality as macrophages, or is that incorrect? Like they need to be activated
I've
before.
seen them phagocytose.
Yeah.
I throw stuff in there and they gobble it up.
Oh, okay.
So again, I'm happy to learn something.
Mm-hmm. For
Like
sure.
learning new things, but I don't fully understand the difference beyond kind of where they're located.
Yeah. No, I agree. I think it's all, there's still a lot to be discerned about macrophages and
Yeah.
monocytes.
And that's, having somebody say that there are different types of monocytes, that doesn't surprise me much because think when we isolate monocytes,
Mm-hmm.
we end up with a mixed population, some that express CD16, which
Right.
is the FC receptor 3, and some don't.
Right.
So it is not a uniform population.
Mm-mm.
And what does one set of those do compared to the other set?
Mm-hmm.
Don't know.
Yeah.
Don't know. So that they're heterogeneous, I buy that. But I guess my question, I would be surprised if somebody found out that they have a very distinct lineage and this type develops into M1 and this other type develops into M2. That those distinctions to me seem to be, they're not immutable. You can take an M2 and turn it into an M1 and vice
Yeah.
versa.
Absolutely. Yeah, for
And
sure.
we sure hope that's true because otherwise we're stuck with these TAMs causing problems for us.
Yes. On that, Jennifer, who had asked a question earlier, had just said loss of CD14 and gain of CD16 is how I've defined whether a monocyte has become a macrophage. Fair.
Oh, well,
Yeah,
I
which is
don't know
true.
about that though. Yeah,
Well, I mean, it's true that I think that a lot of
maybe.
people use that, but I don't know, just like we've talked about, whether just because everything just seems very much on a spectrum, it's hard to discern and say, yes, this is a monocyte. Yes, this is a macrophage at this point. Like,
Right.
yeah,
Because, well, okay, so if it had CD16 and it wouldn't matter that it was in the circulation, you call that CD16 positive thing
yeah,
a macrophage,
I think a lot of the times what
but
I've
it would
seen,
still express CD14.
yes. So I think a lot of the times when people are trying to discern the different populations, rather than just saying monocytes and macrophages, at least when I was still in the lab, we were using the markers as part of the name, like you're just saying, CD16 positive, CD14 negative macrophage versus a double positive versus whatever, to try to
Got it.
get more of a distinctive marker on it. I don't know if
Right,
that means
right.
anything, but yeah. I think it's just with macrophages, even with T cells, but more so that there's still so much left that we don't know, that it just seems very muddy sometimes.
Yeah. Well, that point makes me want to kind of go and look at the cells and say, okay, if I was to separate my monocytes into CD16 negative and CD16 positive,
Mhm.
what happens when I culture those two populations
Yeah. Yeah, what do they become?
in terms of their surface markers?
Mhm.
Yeah,
Yeah, fair. Go, Anne.
interesting.
Go back.
Hey,
Get in the
more
lab.
experiments. Back to the lab.
Let's back to the lab. Um, let's see. We probably have time for a couple more questions here. So, the next one is there any relationship with mitochondrial dysfunction with macrophage development?
Hmm.
You're getting some good
I don't
questions
know much
today.
about What's that?
I said you're getting some good questions
Yeah,
today.
you guys are really trusting me. Don't know much about mitochondrial dysfunctional phenotypes.
Okay, fair
You'd
enough.
think those would be yeah. Don't know enough about that one. Sorry.
Fair enough. Fair enough. Do macrophages stick very, very tightly, even to glass and other substances while culturing?
Was that a question?
Yes,
We could make it a statement because they do
they
attach
do.
very tightly to glass
Yes, and other
and
substances.
plastic
Yes.
and other substances. They even stick to Teflon. So there you go.
Oh, wow. Okay.
I've tried culturing them on Teflon, and you can get them off of Teflon easier, but it's not. They still stick.
Yeah. But kind of like how we were talking about before with them sticking when possibly trying to phagocytose something that they shouldn't be. Doesn't seem like it would be the best culture,
Right,
like
right. It doesn't
environment.
seem like
Yeah.
it, does it?
Um,
Yeah.
so I don't when you start a new macrophage cell line, how many passages do you need to go before you can store some of the cells in liquid nitrogen for future use?
Ooh. Well, don't try doing that with human monocytes or macrophage,
I
for
think they're
one.
saying a cell line, so like THP1 or something.
Oh, well, THP1. See, I don't know. There are changes,
Oh,
aren't there?
they're
And I
person.
think we've usually
What?
tried to limit the time in culture to a month
Okay, culture in a month. So
and then go back to a fresh culture.
it looks like they're using raw 2647 cells, but I suppose
Right,
it'd be the same
right.
thing. Yeah.
I think it'd be about the same thing.
Okay, so about a
I
month.
think I have more experience with THP1 than the raw,
Hmm.
but the THP1s, we were changing them out after a month.
Okay, makes sense. And this should be our last
Yeah.
question here. So we have you ever tried to extract the extracellular vesicles from M1 or M2 macrophages? And then they go on to say, I was thinking to use the extracellular vesicles of macrophages for disease therapy. I could not get enough EVs from microglia, then I want to replace it with blood macrophages.
Oh. Well, we have considered making exosomes
Mm-hmm.
from our cells
Okay.
because there is so much interest in exosomes these days, but we have
Yes.
not gotten around to it. Um, and we would need to characterize them, and because it seems almost too simple to me. Oh, you collect the extracellular stuff, and that's your exosomes. And it's like, no, really?
Yeah, it's got to be more than that.
I would want to be able to tell you something about them.
Right.
So if we do that, you can hear about it.
The next webinar.
um, at the next webinar, we'll talk about exosomes because yeah, that would be an interesting. It would be an interesting product.
It really would. Yeah, I agree. Well, thank you again, Anne, for a fantastic webinar. This was really interesting. Um, and I just want to thank everybody else that has tuned in today. And we will have the webinar on our website in about 24 hours. So again, until next time, good luck in your research. Thank you again for Anne, and goodbye to all of us from Astarte Biologics and Bite Size Bio.
Thanks very much. Bye bye.